We have next speaker is none other than Professor N.G. Prasad. I know him since our PhD days. He did his PhD from JNCSR under the guidance of Professor Amitabh Joshi ji, and he is the founder secretary of this ISEB, and he is very active in this area. And Prasad, please go ahead, please.
Thank you, Sanjeeva. I would like to first thank the organizers of ISEB 5 for inviting me and giving me this chance to talk. I saw that the topic of ISEB 5 was bridging generations, celebrating evolutionary science. So I decided to give a talk on transgenerational effects of infection. So, it's a trait that transcends generations and is important for the evolution of immunity. This work was essentially the PhD work of Abir Basu, who is sitting here. He's now a postdoc and he has a poster up. He's doing some very interesting, very cool work using microbes. Please visit his poster. There are many other students from our laboratory. They're presenting many other posters. Please do visit them. Okay. So let me start by talking about something which is much more general, something called adaptive transgenerational effects. Simply put, if the ancestors experience a certain stressful environment and this increases the fitness of the offspring, then we say adaptive transgenerational effect has happened. Consider a set of ancestors, let's say, shown there in red and gray. A set of ancestors, one of them is experiencing a stressful environment, the other is not. Now take progeny from both these sets of ancestors, subject them to the same stressful environment. If the performance of the individuals whose ancestors experienced a stressful environment is better than the performance of the individuals whose ancestors did not experience a stressful environment, we say adaptive transgenerational effect has happened. Now these effects are not supposed to be genetic. These are supposed to be because of information passed on epigenetically from parents to offspring. But these effects are supposed to be very, very important because there is a certain hypothesis which says that when an organism is presented with a new environment, it is possible that its first response is not genetic, but it is a plastic response, like the adaptive transgenerational effect. And these responses help the population survive the new environment for the initial few generations before the genetic mechanisms kick in and genic capture actually happens, or assimilation actually happens. So if this phenomena is very, very important, the question is how common or prevalent is it? There's a very recent meta-analysis which looked at these phenomena across taxa and across traits, and what they find is most transgenerational effects seem to be adaptive in the sense that the progeny seem to reap a benefit of the parental effects. But how much of the benefit is reaped depends on the trait that is looked at, let's say physiological trait versus life history traits versus reproductive traits. It also seems to be affected by the taxa that you are looking at. Such adaptive transgenerational effects were most pronounced in annual plants and invertebrates. They were less pronounced in vertebrates and were totally absent in perennial plants. Now, one of the subgroups of such adaptive transgenerational effects is transgenerational immune priming. Now, what is transgenerational immune priming? Consider two sets of parents. For one set, you infect them with a heat-kill pathogen or a pathogen that is not very effective, such that you elicit an immune response in the parent, but you don't kill the parent. Right? You take the progeny from both sorts of parents, and then you subject the progeny to infection from the same pathogen. If the performance of the individuals whose parents were infected is better than the performance of the individuals whose parents were not infected, then you say there has been transgenerational immune priming. Right? Now, transgenerational immune priming is right now a very hot area of research, and people have defined typical characters of what you should call as transgenerational immune priming. Three characters are supposed to be very important. One is specificity, long-lastingness of the effect, and a biphasic immune response. If these characters are present, then people say you should call it transgenerational immune priming. If they are not, then you should call it immune enhancement. People agree, people don't agree. For the purpose of the talk, I'm not going to bother about this distinction. I'm just going to use it as it is, right? Now, since this is a hot area of research, there are a large number of studies that look at transgenerational immune priming. This is a small part of a really, really long list of studies from a very recent paper. People have looked for transgenerational immune priming in various kinds of study subjects, starting from crustaceans to Drosophila to bees to mosquitoes, you name it, all those organisms have been used. Large number of pathogens have been used, and people have measured transgenerational immune priming using different measures of immunity, for example, prevalence or viral virulence or the survival of the host or the expression of immune genes, etc. The summary is, when you look at across these studies, some studies find evidence for transgenerational immune priming and some don't. But what is common across all these studies is the following. None of these studies, or most of these studies, do not use infection with a live pathogen in the parental generation. The parents are typically infected with heat kill pathogens or weakened pathogens or their correlates like the LPS or Cephedex beads, etc. Now, this is a good experimental protocol because it allows you to ensure that whatever effect that you see in the offspring generation is not because of survival bias in the parental generation. Also, you can differentiate between the host effects versus the pathogen effects and all that, but it remains that this method is not natural in the sense all the effects of the pathogenicity of the pathogen is lost. More interestingly, there are some studies that have come around recently which say that if you take organisms like beetles and Drosophila and you infect the parents with a live pathogen, then in the offspring generation you do not see any immune priming life effects. It is in this background that we decided to do some studies. And we asked some very specific questions. We asked whether infecting flies with live bacterial pathogen can actually improve offspring immune function. If it does, then how long does this improvement last? How many generations? Is the improvement in offspring immunity specific for the pathogen that was used to infect the parents, or it could be more general? Do the flies that are infected, the parental generation, continue to produce offspring that are more immune throughout their life, or does it happen only in specific windows? And are there any costs associated with these transgenerational effects? Right? So to address the first question, which is, does infecting flies with live bacterial pathogen, does it improve offspring immune function? We took a very simple, straightforward experimental protocol. What we did was we took flies, Drosophila melanogaster, and we divided them into two groups. One was the infected group, the other was the control group. This was the parental generation. The infected group was infected with a gram-positive pathogen, Enterococcus faecalis. Right? 48 hours after infection, this number is important. 48 hours post-infection, we collected eggs to start the next generation. When those adults grew up, they became the offspring generation. In the offspring generation, again, each of these were divided into two groups. One got infected, the other was sham infected. We again collected eggs from them to start the grand offspring generation, where again, one group was infected, the other was not infected. What is important to notice is Enterococcus faecalis was the pathogen for the parents, for the offsprings, and the grand offsprings, and the eggs were always collected 48 hours post-infection. What did we find? Well, if you look at the parental generation, what you find here on the x-axis is the time post-infection, on the y-axis is survivorship probability. The colors and the line schemes will remain the same throughout all the graphs. The solid lines will be for females, the dotted lines will be for males. The purple color will be for the control and the green color for the infected. So if you look at this, with sham infection, there is very little mortality in the parents. With actual infection, there is a lot of mortality. About 50% of the parents die, 50% survive. Mortality is rapid in the initial phase, and it sort of plateaus about 48 hours post-infection, and it is in this 48-hour window, what is colored red is we collect eggs to start the next generation, right? So some survive, some die, we collect eggs from the survivors to breed. What happens if you look at the offspring and the grand offspring generation? Well, the first graph shows you what happens in the offspring generation. Remember, the offspring generation has been infected with Pseudomonas entomophila or is sham infected. Now, if you look at the females, the individuals whose parents were infected survive much better than the individual whose parents were not infected. If you look at the males, again, individuals whose parents were infected survive much better than the individuals whose parents were not infected. So very clearly, if the parents were infected, the progeny reap a benefit in terms of surviving the same pathogen better. There is clearly a transgenerational parental effect of infection. What about in the grand offspring generation? Well, the effects sort of decay. If you look at the females, again, individuals whose Can you hear me? Yeah, okay. Individuals whose parents were infected do much better than the individuals whose parents were not infected, but the magnitude has gone down. But if you look at the males, there is no such effect. So what does this tell us? Well, there is certainly a transgenerational effect of parental infection, but this effect is not long lasting. You see it very clearly in the offspring generation, but it seems to decay by the grand offspring generation. Why is this important? Because if such plastic effects were very important in helping the population survive in the initial few generations, at least it's not very important in our case. Using the host that we have used and the pathogen we have used, this effect seems to be very short-lived, and therefore it is very unlikely to affect long-term evolution of immunity. Right? How does the progeny generation survive the pathogenic infection? When you take an organism and infect it, organisms typically have two different ways in which survive infection. One is to restrict the pathogen load, what we call resistance. The other is to maintain higher fitness even though the pathogen load is high, what we call as tolerance. Now, tolerance is very difficult to measure, so we decided to measure resistance and asked, does resistance play a role at all? So we did an experiment where we took the parental generation. The parental generation was either infected with Enterococcus faecalis or was sham infected. We took progeny from them, and in the progeny generation, again, the individuals were infected with Enterococcus faecalis or were sham infected. 4 hours and 10 hours post-infection, we extracted the pathogen from the body of the flies and quantified how much of the pathogen do they carry. Now here are the results. If you look at the female offspring, 4 hours post-infection, there is no difference in the bacterial load of those individuals whose parents were infected and the individuals whose parents were not infected. However, 10 hours later, the individuals whose parents were infected have significantly lower bacterial load compared to the individuals whose parents were not infected. If you look at the males, the results are identical. So what this tells us is that if an individual's parent had been infected, then that individual is better able to resist pathogenic growth within their body and therefore survive infection. Resistance clearly plays a role in the survival of the offspring generation. We then asked, okay, offsprings can survive infection better if the parents have been infected, but what if we change the pathogen? What if the parents are infected with one kind of pathogen, whereas the progeny are infected with a different kind of pathogen, right? Do they still maintain the survival advantage? So in the parental generation, we infected the flies with Enterococcus faecalis, but in the progeny generation, we infected the flies with different pathogen, which is Bacillus thuringiensis, Erwinia caratovora, or Pseudomonas entomophila. So then we ask what happens. Well, if you look at it, the first graph is for progeny that were infected with Bacillus thuringiensis. Remember, their parents were infected with Enterococcus faecalis. We really find no advantage of survival, whether the parents were infected or not infected, the progeny all die at the same rate. So clearly there is no survival advantage. If you look at progeny that were infected with Erwinia carotivora, if you look at the females, individuals whose parents were infected do much better than individuals whose parents were not infected. Same results for the males also. So when the parents are infected with Enterococcus faecalis, progeny survive infection with Erwinia carotivora better. If you look at progeny infected with Pseudomonas entomophila, again we find no evidence of any survivorship advantage. So what does this mean? Well, specificity has been considered as one of the hallmarks of this transgenerational effect. Our results show that this is not necessarily the case. Does this mean that we find a general response? No. It seems to be a little more nuanced. When the parents are infected with Enterococcus faecalis, progeny survive infection better with Erwinia carotivora, but not with Bacillus thuringiensis or Pseudomonas entomophila. So there is some generality, but not complete generality. Therefore, it is certainly not very specific. That is very true. The second thing that this result shows is that this result is against a very long-held notion about Drosophila immunity. People typically say that when flies are infected with a gram-positive pathogen, then your toll pathway kicks in, and if it is gram-negative, then the IMD pathway kicks in. The parents in our experiment were infected with a gram-positive pathogen and the progeny with a gram-negative pathogen, yet the progeny reap a benefit, which means that there is certainly considerable crosstalk across these pathways. Now we wanted to ask a very important question, which is, do infected parents produce more immune progeny throughout their lives, or does this happen only in certain windows? To address this question, we repeated the experiment that we did in the beginning with one major difference. Remember the experiment? You have the parental generation from which the offspring generation is generated, from which the grand offspring generation is generated, and in each generation you infect those set that is to be infected with Enterococcus faecalis. The only difference between this experiment and the previous one is this. In the initial experiment, remember that post-infection, in a window of starting at 48 hours, we collected eggs to start the next generation. Whereas in the current experiment, we moved that window from 48 hours to 96 hours. So in this experiment, when you infected, some individuals would die, some individuals would survive, and the survivors would lay eggs in a window starting at 48 hours, and those would contribute to the next generation. Here, again, some would survive, some would die, and the survivors would lay eggs starting at a window of 96 hours, and those would contribute to the next generation. That is the only difference, right? What are the results we find? Well, if you look at the offspring generation, if you compare the two dotted lines and the two solid lines, there really is no significant difference. So even in the offspring generation, we do not see any benefit of parental infection being reaped by the progeny. If you look at the grand offspring generation, again, we do not find any benefit of parental infection. What does this tell us? Well, that parents produce immune progeny only in those windows that are closer to infection. If you collect eggs from a window starting at 48 hours, the progeny tend to be more immune. If you collect eggs from a window that is 96 hours post-infection, progeny are not immune. Why does this happen? There have been many hypotheses proposed. One very interesting hypothesis is that parents probably produce and transfer some costly molecules as indications of immunity to their progeny. If this molecule is really costly, then you would not expect the parents to produce it constitutively. Therefore, maybe they produce it only in certain windows. I'll not take the third one in the interest of time. Okay. We then ask two very important questions. One, do the progeny pay a cost of improved immune response. It has been suggested that when the parents are infected, the progeny become better at surviving infection, but they pay a cost in some other trait. Is this really true? So we took parents, infected them, took the progeny. Without infecting them, we looked at the reproductive performance. For females, we looked at fecundity, and for males, we looked at competitive fertilization success. What we find is there is no significant difference in the fecundity of individuals whose parents were either infected or not infected. In the males, there is no significant difference in the competitive fertilization of those individuals whose parents were infected or not infected. So there is no cost that is being paid by these individuals that are more immune. Finally, we asked, if you infect the parents, take the progeny, and infect the progeny, they do better in terms of survival under infection. What about other traits under infection? What if I looked at, let's say, fecundity? Will their fecundity be much better? So we took infected parents, took the progeny, and the progeny were subjected to either sham infection or infection, and then we looked at their fecundity. Under the sham infection condition, we did not find any significant difference in fecundity. Under the infected conditions, we did not find any significant difference in fecundity. Clearly showing that whatever the benefits that the progeny accrue is limited only to survivorship and does not extend to other traits. Okay. So let me just put up the summary and end it here. Thank you.
Thank you, Prasad. We can take one or two quick questions, please. Yeah.
Hi. I was just wondering what is the background of the flies on which this experiment is done. The reason I ask that is we know partly from your earlier work that parental environmental effects, not in those studies it wasn't infection but parental age, parental larval or adult densities, etc., can result in the evolution of very different patterns of parental effects in different selection regimes.
Correct.
So
these flies are baseline populations. These are the Blue Ridge baseline populations. These are 14-day discrete generation cycle populations. They've been in the lab now. When we did the experiments, they were in the lab for nearly 10 years plus under standard conditions. So they are not consciously under any selection. So these are background flies. We have tried it with some other populations. Again, the results are highly variable depending on the population and the pathogen. Sometimes you see it, sometimes you don't.
thanks.
Yeah, one more question here. Yeah, she has right the question.
Hello, thank you for the talk. Now, I would like to ask if a similar response can be seen in any organism, like in your experiment, we see that the response was seen for an infection. So, could you say that a similar response might be seen for as a response to a chemical contaminant in the environment?
Yeah. So parental effects are ubiquitous. I mean, you can find it for all kinds of things, right? You could find it for maybe a chemical contaminant, you could find it for some heat stress, cold stress, all kinds of things are possible. In fact, many of the times when we do selection experiments and we do assays on selection experiments, this is something that we have to be very, very conscious about. Do you want to look at only the genetic responses, so to call inherited responses, or do you also want to look at the responses of this kind which are not strictly genetic in that sense? So we have to take care to ensure that such parental effects are normalized across your selected and control populations before you do experiments. So short answer is yes, they are known to be ubiquitous. You can find it in many, many, many different conditions.
Thank you.
Yeah, you wanted to ask question? Yeah.
Okay, so very quickly, you must have speculated about what the chemical is that might be passed between the generations, whether it's part of the pathogen like a human vaccine or something else. So what are your thoughts on that briefly?
I mean, other people have speculated about what this is. One speculation has always been it's maybe a part of the pathogen that is packaged into the eggs and passed on. But that would not really explain why the signal should be costly. So another hypothesis has been that it's something that the host itself produces. It need not necessarily be something very unique. It could be a combination of what is normally produced. But what is very clear is, in some of these experiments, you see incredible specificity in terms of immune priming. One strain of, let's say, Bacillus thuringiensis will elicit it, and if you infect the pathogen, progeny with a different strain of the same bacteria, you will not find an advantage. So in some cases, there is something that also conveys information about the identity of a strain. People have been looking for it, people have not really found it. One thing I will add to it, this is, we have found parental effects through the mother, but we have also found it through the father, where explaining it becomes a little more difficult. Yeah.